Liebe Beatrix, lieber Uli, liebe Mitglieder der Georg Friedrich Götz Foundation, it is an honor to be here today, and if you don't mind, I switch to English because I see a couple of non-German speaking scientists in the audience, and I would love to share my research with everyone. As I said, it is my distinct pleasure to share with you some of my previous work, and particularly today, I will talk to you about this study, Unleashing Cell-Intrinsic Inflammation as a Strategy to Kill AML Blasts. So, as you know, all blood cells originate from hematopoietic stem cells, and rarely a combination of genetic events and altered inflammation can drive the transformation of malignant hematopoietic cells. While dysregulation of innate immune signaling is a hallmark of hematologic malignancies, we have very limited understanding of how inflammatory programs change when hematopoietic cells undergo malignant transformation, ultimately resulting into acute myeloid leukemia. AML is the most common malignant myeloid disorder in adults, and it remains poorly treated. Here you see some survival curves from our patients treated here in our department in Zurich over the last 14 years, and particularly in the elderly patients where we know that inflammatory programs play an even greater role, the outcome is dismal. Very simplified, leukemia cells are derailed immune cells, so they are an ideal model to study altered inflammation in malignancies. So we set out to dissect leukemia cell intrinsic inflammatory signaling to discover AML vulnerabilities. Across large cohorts from patients with AML, we identified this cluster of patients with an enrichment for immune and inflammatory pathways. And they always represented about 40% of all leukemia patients, or a relevant amount of patients, and they mainly had a monocytic and myeloid, so driven leukemias. So, and within those patients, using a number of genome-wide CRISPR screens, we discovered interferon regulatory factor 2 binding protein 2 as a selective inflammation modulating AML dependency. So when we then went ahead and validated this target, here you see an experiment where we transduced leukemia cells with CRISPR guides against this target, we found a significant reduction in survival in those leukemia cells, we were able to confirm this in a number of orthogonal models. Here, for example, using RNAi technology in an in vivo study where we transplanted mice with luciferase leukemia cells and an induced knockdown of IRF2BP2 and found that the mice with knockdown of IRF2 had significantly lower leukemia burden. Then we next wanted to determine whether there could be a therapeutic window between targeting IRF2BP2 in AML cells versus normal hematopoietic progenitor cells. And because of the low efficacy of lentiviral transduction, we here used nucleofection to knock out IRF2BP2 in CD34 positive, so healthy hematopoietic stem cells. And you can already appreciate on the Western blot, if I can get this in the middle, never mind, that we got very good knockout of IRF2BP2, and while there was no reduction in colony-forming capacity in the normal bone marrow cells, where in cells from patients, so patient-derived cells, there was a significant reduction in colony-forming capacity. To next assess the effects of IRF2BP2 perturbation, or here loss in patient-derived cells, we transduced cells that we had gained from patients and then passaged a couple of times through mice to get stable Cas9 expression with inducible guides against IRF2BP2. And then we transplanted mice either with leukemia cells with a guide against IRF2-BB2 or a control guide, and then upon confirmation of engraftment, those mice were fed a doxycycline-containing diet to induce knockout of IRF2-BB2. In bone marrow aspirations of those mice, 2 weeks post-knockout of IRF2-BB2, we found a significant reduction in leukemia burden, shown here on your right in this plot. And this translated in a significantly prolonged survival in mice that had leukemia with loss of IRF2-BB2. When these mice eventually died, they died because they had regained expression of IRF2-BB2. So, of course, I was most excited to learn why IRF2-BB2 was so essential for leukemia cells to survive. So, to study this best, we developed a degradation system which allowed us to study acute effects of loss of IRF2-BB2 protein in a way where we knocked out endogenous IRF2-BB2 and overexpressed an exogenous version of IRF2-BB2 with a tag that then, if you add a D-tag molecule, this protein undergoes proteasomal degradation. And here you can see that when testing those cells, again here in a leukemia cell line, there was loss of IRF2-BB2 as early as a few hours after exposure to this D-tag molecule, and this again resulted in a significant reduction in cell viability, confirming our previous observations. We then used this model to perform gene expression studies where we looked at differentially expressed genes only after a few hours of degradation of IRF2-BB2. And what we found was that there were significantly more genes increased in expression, suggesting to us that IRF2-BB2 acts as a transcriptional repressor. We were then also interested to see whether genes that were differentially expressed after perturbation of IRF2-BB2 were actually bound by IRF2-BB2. And when we performed, and this is the shortened result here, chromatin studies, we found that actually genes that we found to be decreased in expression were also mainly bound by IRF2-BB2. We then took all those genes that we defined as IRF2-BB2 target genes, so decreased in expression and bound by IRF2-BB2, and performed gene set enrichment analysis. And what we found was a significant enrichment for TNF-alpha signaling via NF-κB, suggesting to us that IRF2-BB2 represses TNF-alpha signaling via NF-κB. So if repression of TNF-alpha signaling would be the most biological relevant function for this observed dependency, then super-repression of NF-κB should rescue the phenotype. So in a proof of principle experiment, we expressed a so-called super-repressor mutant allele of IKB-alpha, where we mutated one base so that it can no longer be phosphorylated by the IKK complex. And what we found indeed that cells that had this IKB-alpha mutant, you can see this in the very last spot in this Western blot, supported loss of IRF2-BB2 very well. And then when we assessed cell viability, the reduction in cell viability was entirely rescued. So in summary, this study discovered IRF2-BB2 as an AML inflammation modulating dependency. We found that IRF2-BB2 acts as a repressor of TNF-alpha signaling via NF-κB, and maybe most importantly established this concept of cell-intrinsic hyperinflammation as a mechanism to induce apoptosis in leukemia cells. With this, I want to thank you for your attention. I want to thank all my previous mentors, including Markus, who is here today, my entire department, many collaborators, previous lab members who contributed to this work, my current lab, current funding institutions, of course, all patients and families who supported our research, and the UZH Foundation and the Georg Friedrich Götz Selection Committee for choosing my work. Thank you very much.